Hello, I'm Sydney Miller and I'll be focusing on the introduction. So, basement membranes are present in every tissue in the human body, or in animal tissue to be more specific. They provide signaling cues that regulate cell behavior and function, such as in tissue genesis and homeostasis. It is a very thin membrane composed of glycoproteins. The basement membrane separates the epithelium from underlying tissues. We call the basement membrane a specialized type of extracellular membrane, or ACM for short. Here I have some images where you can see the very thin layer, the basement membrane, or short, they also call it epidermal dermal junction. Basement membranes also act as scaffolds for cell migration and adhesion. They offer functional input to monitor cellular function. Collagen is a protein that is most abundant in the human body. The specific type in this lab that was studied was type 4 collagen, and it's most abundant in basement membranes. The recent discovery in this lab of these type 4 collagen scaffolds were in bovine and mice and their tissues, and they found that the protein was stabilized by sulfolamine chemical bonds. So, by studying this, they tried to compare it with the evolution of tissue and the Eumetozoa.
This study began by collecting tissue samples from various animal phylums from different places over the U.S. In order to purify and isolate collagen 4, they froze these tissues and pulverized them with a mortar. The tissues are then placed in a salt solution and spun for 15 minutes. The liquid solution is then put into gel filtration chromatography. The proteins were then stained to analyze what type of collagen 4 is the most common variant out of the tissue samples collected. Then they found the RNA sequencing by using the BLAST databases. They denatured animal NCL hexamers by boiling them and separating them into polypeptides. The polypeptides were then pulled together and freeze-dried. These polypeptides were then mass analyzed in LTQ Orbitrap XL or LTQ Orbitrap Vicryl mass spectrometers to identify the cross-linked peptides. Raw data files were manually searched for by using Excalibur 2.2 browser software or the UniProc KB database.
Hi, my name is Jasmine Morton, and I'll be doing the materials portion of the video presentation. So when I was reading the materials part of the laboratory report that was posted on Blackboard, I noticed that there were a lot of materials that were used throughout the duration of this experiment. It seems as if the scientists went all over the United States just to look for what they were or what they needed to in order to properly conduct this experiment. From LA to Kansas City, a specimen was collected from basically all over the United States. So various objects and cultures were used, such as various strains of cultures of epithelial tissue, marine life, such as Hydrotina polycina, and Ectopleurolarynx. Sorry for my mispronunciations, but all of these materials were either purchased from private laboratories or public laboratories, national associations, or donated by generous people of the scientific community. Two people that were mentioned were John and Lisa Fuller, and they were obviously donated, or generously donated, D. melanogaster, as well as a mouse monoclonal antibody, and these two people are professors from the University of California. One thing that came to my surprise as I was reading the material list was human kidney tissues. These were generously obtained by the National Disease Research Interchange. Again, the human donors were aware of what their organs were going to be used for, which was nice of them to do so. But in order for them to actually obtain human tissue, it's kind of hard to do so. You have to be kind of notable in the scientific community. So in order for these researchers to even be considered, you know, worthy in a sense of having these types of important tissues at that. That allows one to believe that these scientists and are very serious about their experiment. Again, since there was so much diversity in what they had with their materials, such as marine life, plant life, and even human tissue, it allowed them to have different types of diversity. Instead of just restricting themselves to one specimen, they were able to look at a bigger picture of various types of tissues here on Earth.